Hi, my name is Alexis Pratt. I'm a senior biology major at Birmingham Southern College, and today I'm going to be discussing the research I completed in collaboration with Hudson Alpha's Code Project. So, myosin-binding protein C is a thick filament sarcomere protein that regulates muscle contraction and is essential for cardiac muscle function. Mutations in the gene MYBPC3 that encodes for this protein account for more than half of the mutations that cause hypertrophic cardiomyopathy, which is a very prevalent heart disease that consists of the abnormal thickening of the heart. About 1 in 500 people, specifically young people and athletes, are affected by this disease. Given the increasing technological advances in genome sequencing, an overwhelming number of genetic variants or mutations are being detected with genes such as MYBPC3. A majority of these variants are being classified as variants of unknown significance, VUS, because of their impact on the function of the health of an individual is unknown. These novel variants can differ across each individual and are important to research and characterize for patient-specific therapy and treatment. My research involved analyzing a suspected pathogenic VU in MYBPC3 and providing evidence for how it might affect the structure and function of the myosin-binding protein C. Specifically, I looked at the structural impacts of the mutation of tryptophan to arginine at position 486 has on the protein. This mutation involves the substitution of a charged hydrophilic residue for a neutral hydrophobic residue. Given the opposing properties between the residue's side chains, it was expected to observe structural changes in the protein. The first part of the study involved curating MYBPC3's variants, specifically the VUs. This was done using the web-based software ClinVar Miner and Ensemble. Ensemble provided various measures of variant deleteriousness such as CAD, polyphen, and SIFT algorithms which were used to choose a likely pathogenic VU. Provian, a variant effect analyzer, provided similar information for determining a variant's deleteriousness. Further analysis of the position in the protein's amino acid sequence that my VU resides at then took place. I specifically investigated the sequence conservation and homology of the wild-type tryptophan at position 486 using the conserv server and Cluster Omega. Next, I was able to characterize MYBPC3's function and interactions with other proteins using the string database. Lastly, 3D modeling and structural analysis were incorporated to investigate any structural changes the mutation may have caused on the wild-type protein using ITASR, VMD, HOPE, and 3D ligand site. The data obtained from Provian predicted that the tryptophan at position 486 to arginine variant is deleterious, which suggests that it might be pathogenic. The conserv data revealed that the tryptophan at position 486 is buried within the protein and is highly conserved across species. Likewise, the Cluster Omega multiple sequence alignment confirmed that tryptophan at 486 is highly conserved across 10 analyzed species. This suggests that this position is of structural or functional importance for MYBPC3. The string database revealed that the protein interacts with various other sarcomeric proteins including myosin, titin, troponin, and tropomyosin. Given that MYBPC interacts with all of these proteins suggests that these interactions are a crucial part of its function and is what aids the regulation of muscle contraction. A predicted protein structure of MYBPC was obtained from ITASR and was then mutated in VMD. The 3D modeling in VMD revealed that the mutation results in structural changes such as the lengthening and deletion of beta strands and beta turns, which are structures that connect the beta strands together in various regions of the protein. "3D ligand site predicted that the mutation introduces new ligand binding sites compared to the wild-type protein. The function of the domain that my VU resides in is unknown, but it is speculated to serve as a stabilizing point for the flexibility of the surrounding 10 domains in the protein to interact with myosin. Therefore, these structural changes are predicted to affect those binding interactions. In conclusion, the structural changes that occur when the highly conserved residue tryptophan is mutated to arginine suggest that the VU is pathogenic. This VU likely disrupts the hydrophobic interactions at the core of the protein, resulting in functional degradation such as its numerous protein interactions, and potentially leads to the onset of hypertrophic cardiomyopathy. Characterizing VUs such as the one I researched is a crucial area in research that can provide clinicians further insight on how to diagnose and treat patients with VUs. Thank you so much for taking the time to view my poster. If you have any questions or comments, feel free to leave them down below or contact the email that I have provided on the bottom of the poster. Have a great day.